Hello students, welcome again to the channel Physiology Redefined. So today I will talk about the monophasic and the biphasic action potential. So in the last few videos, I explained what is action potential. So there you can see that if two electrodes are placed on the cell membrane, one is in the outside, another is in the inside, and those two electrodes are connected to the voltmeter or cathode ray oscilloscope, then a unidirectional action potential can be recorded. Unidirectional, that is in one direction only. So that type of action potential we talked about in last few videos, that is known as the monophasic action potential, because this is in only one direction or unidirectional. So let us see in details how these monophasic and the biphasic action potential can be recorded and what are its importance. So I would request you to watch this video till the end. So first is regarding the monophasic action potential. Monophasic action potential. So here you see, this is suppose a cell membrane, outside of the cell membrane, inside of the cell membrane. Now if one electrode is placed in the outside of the cell membrane and another electrode is placed at the inside of the cell membrane, and these two electrodes is connected with the cathode ray oscilloscope. Then what's happened? A action potential will be obtained. This is in duration, in millisecond, and this is in potential difference in millivolt. So if a stimulus is given, in response to the stimulus, you know the action potential is initiated. And what type of action potential can be recorded in the cathode ray oscilloscope? That is the unidirectional action potential. Unidirectional action potential will be obtained, and in this action potential, this is the depolarization, this is the rapid depolarization, this is the late repolarizations or slow repolarizations or after depolarization, and this is the hyperpolarization. So these are all this is the phase 1, phase 2A, phase 2B, phase 3, and this is again the resting phase or phase 4. So all of the phases of a single action potential recording that can be obtained if two electrodes are placed on the cell membrane. One is in the outside, another is in the inside. Now same type of action potential can also be obtained. So this is the cell membrane, outside, inside. In this case, what's happened? Both the electrodes are placed in the outside. But one electrode is placed at the healthy side of the cell membrane, and the another electrode is placed at the injured side of the cell membrane. So in this case, unlike this one, so this is OR. Unlike this one here, two electrodes, one in the outside, another in the inside. But here, both the electrodes are placed in the outside of the membrane. One is at the healthy side, normal side of the cell membrane, and another is at the damaged side or the injured side or the sectioned side of the cell membrane. In such a case, what's happened? The action potential that can be recorded by the cathode ray oscilloscope will also be the unidirectional. That is unidirectional, and this type of action potential is known as the monophasic action potential. So this is the monophasic action potential, which can be obtained by placing two electrodes. One is in the outside, another is in the inside, or two electrodes, both are in the outside, but one is at the healthy side, another is at the damaged side of the membrane or the injured side of the cell membrane. In such a case, a unidirectional action potential can be recorded, and that is known as the monophasic action potential. So monophasic action potential is the unidirectional. This is unidirectional. And this is also the intracellular recording. Intracellular recording. Because in this case, the inside of the cell membrane are getting connected. In this case, it is inserted in it is pierced, second electrode is pierced through the cell membrane, and thereby intracellular recording. In this case also, as cells are damaged, cell membranes are damaged, there is no need to pierce it, though no need to insert the electrode in the inside, but it is getting contact with the intracellular fluids, and thereby it is the intracellular recording. And this type of action potential has got little clinical importance, little clinical importance. Now regarding the biphasic action potential. So what is biphasic action potential? So from the name it implies, biphasic means it is bidirectional. So action potential can be recorded both in the upward, as a upward deflection followed by the downward deflection, positive direction and the negative directions. Here it is in the positive direction or upward deflection. So in this case, monophasic one is the upward deflection. So next one is the biphasic action potential. Biphasic action potential. What is this biphasic action potential? So biphasic action potential is the action potential which can be recorded if two electrodes are placed on the cell membrane. So here you see the cell membrane outside and the inside, and both the electrodes are placed in the healthy side of the cell membrane. Both the electrodes are placed at the healthy side of the cell membrane to the cathode ray oscilloscope. So if both the electrodes are placed on the outside of the cell membrane and cell membrane where the electrodes are placed are healthy, not injured, not damaged, not sectioned, in such a case what's happened? This type of action potential will be obtained. So this is duration in millisecond, and this is the potential difference in millivolt. So in such a case, if a stimulus is applied, then what's happened? The biphasic action potential will be obtained. Biphasic action potential will be obtained, And in such a case, the record will be the positive directions followed by a isoelectric period, and then the negative direction. So this type of cell is the upward deflection. This is the downward deflection. Followed by the and here is the in between there is a isoelectric period. So in between two upward deflections of the action potential, upward and the downward deflections of the action potential, there is a very short isoelectric period. And as in this case, the action potential is recorded in both the direction. It is known as the biphasic action potential. So in this case, what's happened? It is the bidirectional recording. And this is in this case entirely extracellular recording, because here you see both the electrodes are placed on the extracellular surface of the cell membrane. So it is the extracellular recording, and in this case the upward action potential will be recorded as upward deflection followed by isoelectric period, followed by the downward deflection. And this type of recording of the action potential is very, very important clinically. So it is clinically more important. This recording is very important, because all the electrical recordings that is used for the clinical diagnosis of the different systems are under this category. For examples, the ECG. Examples are ECG, EEG, EMG, electroretinograph, so electroolfactogram. All these are the recording which are the examples of the extracellular recording and which are used to diagnose any abnormality of the underlying tissue. For example, ECG, from the ECG we can get the idea about the status of the cardiac systems. For the EEG we can get about the idea regarding the status or normal functionings of the different parts of the brain. So this is why this type of recording, extracellular recording, biphasic action potential, is clinically much more important. So this is all about the monophasic and the biphasic action potential. Now there is another type of action potential which is known as the compound action potential. Compound action potential. So what is this compound action potential? Another type of action potential that is known as the compound action potential. So let us see now what is compound action potential. So this type of action potential that I discussed just now, monophasic and the biphasic action potential, these can be recorded from a single cell membrane or from a single neuron. But if the stimulus is given to a nerve trunk which is comprised of a collection of the nerve fibers, that means if a stimulus is given to a mixed nerve and action potential is recorded, then what we can see, a different type of action potential can be obtained. So in this case, compound action potential is the action potential in mixed nerve fiber which contain different neuron of varying diameter. A nerve trunk of the mixed category can contain different type of nerve neurons with varying diameter. Now, if such a mixed nerve is stimulated, then what's happened? This type of action potential can be obtained. So, duration in millisecond, electromotive force or potential difference in millivolt. So, if a stimulus is given here, then what type of action potential can be recorded? There is a action potential of OAV nature. So this type of OAV, multiple peak of the action potential, can be recorded, and that this type of action potential is known as the compound action potential. So what for this type of OAV pattern appears? This is because, as I told, mixed nerve contains different neuron with varying diameter. For examples, as per the varying diameter is concerned, the nerve may contain the three category of the nerve neurons. First one is known as the group A neurons. Second one is known as the group B neurons. And third one is known as the group C neurons. There are three categories of the neurons depending on their myelinated property. Generally, the A type of neurons are heavily myelinated. So, as you know, neuron has got some insulation, layer of the insulation, that is known as the myelin sheath. So, these myelin sheath are much more in diameter in case of the A category of the fibers. So regarding these details of these fibers, I will discuss in another video under the neuromuscular system. Now, this A category of the neuron is comprised of four type of fibers, four type of neurons. One is known as the alpha, another is the beta, third one is the gamma, and fourth one is the delta. That means A are further classified into A alpha, A beta, A gamma, A delta. Diameter of the A alpha is much more than the B. B's diameter is more than the gamma, and gamma's diameter is more than the diameter. So as the diameter varies, what's happened? The nodes of radivia are distantly located, are far away from each other. And as I told in another video, that in case of myelinated nerve fiber, the action potential is jumped from one node to the another node, which are distantly located. So in this case, for example, A alpha, myelin sheath diameter is much more. So nodes of Ranvier are distant, located far away from each other. So there is a more jumping of the action potential from one node to the another node, and that will cause the first appearance of the A alpha wave. So this is the wave for the A alpha. So A alpha wave will appear first. A alpha wave will appear first because its diameter is much more, and thereby the appearance of the action potential will be much more. Then this is the curve, this is the peak, this is the curve which is known as which is because of that or due to the A beta neurons or nerve fibers which are having less diameter in comparison to alpha, and thereby action potential, rate of travel of the action potential will be comparatively slower, and thereby their peak will appear after the appearance of the A alpha. In this way, this is the curve or the wave for A gamma, and this is the curve for A delta. So all these are the types of the A category of the fibers, A type of the fibers, which are highly myelinated. Now this is the type of the fiber which is type of the wave which is due to the B fibers. So as I told, B fibers. So these are all the heavily myelinated, and this is the fiber which is mildly myelinated. So as it is mildly myelinated, diameter of the myelin sheath, thickness of the myelin sheath is comparatively less. So nodes of Ranvier will appear within a short distance, and thereby their rate of travel of the impulse will be slow, and as the rate of travel of the impulse will be slow, this will appear late, and this type of wave is known as is due to the B category of the fibers. And lastly, C fiber is this is the curve, action potential curve for the C category of the fibers, which is unmyelinated fibers. So as it is unmyelinated fibers, neurons are unmyelinated, then what's happened? No nodes of Ranvier. So action potential has to pass through each and every part of the cell membrane, every part of the neuronal membrane, and thereby they are slow. Their passage of the impulse will be slow, rate of the impulse will be slow. As these are slow, it will appear late, very late, and this type of action potential is because of the C category of the fibers. So if the mixed nerve having all the nerve fibers are present, then we can see a wavy pattern of the action potential, and that type of wavy pattern of the action potential is known as the compound action potential. So compound action potential is the wavy pattern of the action potential, due to presence of multiple neuron, multiple nerve fiber, multiple type of nerve with varying diameter, with different diameter. So this type of action potential is known as the compound action potential. So this is all about the most important forms of the action potential, common forms of the action potential, that is the biphasic action potential, monophasic action potential, and the compound action potential. The details of these things will be in the description box, so you can get some details of this action potential, of this type of action potential in the description box. I hope it is very clear to you. If you like this video, don't forget to share it, subscribe it, and press the like button. Thank you.